Good afternoon, everyone. We are Nara and Alora, and we are going to present article titled "Quantitative Proteomics of Hamster Lung Tissues Infected with SARS CoV-2 Reveal Host Factors Having Implication in the Disease Pathogenesis and Severity." The paper published in the FASEB journal at May 2021 by Suresh V, Mohanty, Avula, and their colleagues in India. The aims of the study are to provide a deeper understanding of the host response to SARS-CoV-2 infection in hamster lung tissue and to quantitatively compare the lung proteome in SARS-CoV-2 infected hamsters at various days post-infection, identifying differentially expressed proteins. As you are all well aware, the outbreak of the coronavirus known as SARS-CoV-2 has unleashed a worldwide crisis affecting millions of individuals across the world. This virus is the causative agent of the COVID-19, primarily targeting the respiratory tract, often leading to respiratory failure reminiscent of acute respiratory distress syndrome (ARDS). A hallmark of this disease is an aggressive inflammatory response resulting in substantial tissue damage and, in severe cases, fatal lung injuries. The interplay between the virus, epithelial, endothelial, and immune cells lead to a dysregulated inflammatory response that exacerbates respiratory distress and disease severity. After identifying the SARS-CoV-2 virus, multiple genomics and proteomics-based approaches have been adapted to understand the host protein response in this viral infection. Many studies have been conducted using proteomics technology to understand the virus host protein interactome, changes in host protein expression upon virus infection, and for diagnosis of SARS-CoV-2 infection. Hamsters have used for studying SARS-CoV-2 infection, while hamsters mimic a mild pattern of human disease and they do not develop diffuse alveolar disease, a feature seen in severe COVID-19 patients. Some of the differentially expressed proteins identified in this study can also be validated in easily accessible patient samples such as body fluids as potential biomarkers for predicting the disease course.
Now, onto our methodology. Of course, we have to start with the animal ethics. The study was conducted on Syrian golden hamsters and utilized them as a model for the infectivity of SARS-CoV-2. This was approved by the Institutional Biosafety Committee and Animal Ethical Committee of the Indian government, as well as adhered to the specific guidelines in regards to that. For the animal studies, 11 hamsters were acclimatized for 4 to 6 days in the laboratory. 6 of them were infected with the SARS-CoV-2 and 5 mock-infected, where the virus was isolated from local COVID-19 patients. They were sacrificed on the 4th day post-infection, which was 3 of the hamsters, and the 14th day post-infection was another additional 3 of the hamsters. For sample collection, tissues were collected from vital organs such as the lung, the pancreas, and the spleen, and formalin for histopathological analysis, triazole for RNA isolation, and snap frozen samples were kept for further use. Sections were treated for antigen retrieval and peroxidase blocking for the immunohistochemistry, and they were incubated with specific antibodies washed and developed with DAB substrate. Sections were also treated for antigen retrieval and serum blocking for immunofluorescence study, and they were incubated with specific antibodies washed and visualized using confocal microscope. With our Western blot analysis, the cells were lysed in RIPO buffer, subjected to SDS page, and transferred to nitrocellulose membrane. They were immunoblotted for SARS-CoV-2, nucleocapsid protein, and beta-catenin. For the qRT-PCR, RNA isolated from the hamster tissue samples were then quantified of viral load using specific primers and probes for SARS-CoV-2 and gene. And for our proteomics analysis, the lung tissues from the infected and mock-infected groups were processed and homogenized. For our LC-MS-MS analysis with TMT labeling was utilized for relative protein quantification. For our bioinformatics and statistical analysis, protein identification and quantitation using Proteome Discoverer 2.3 was utilized. Data was searched against Syrian golden hamster protein through UniProt, and proteins with greater than 1.5-fold or less than 0.67-fold expressions were considered differentially expressed. But for the statistical analysis, the Venn diagram for comparison of differentially expressed proteins was used, as well as multiple software such as Panther classification, Go, and K pathway analysis. PCA and heat maps were used for data visualization, which you will see in the results.
For the clinical features, at the 4 DPI, it is observed a significant weight loss in all infected hamsters with approximately a 15% of reduction in their initial body weight, which is seen in figure B. After 6 DPI, infected hamsters began regaining their body weight, and throughout the experiments, no mortality occurred in either the infected or mock-infected groups. At the time of organ isolation, it's noticed the gross lung congestion in infected hamsters at 4 DPI. Approximately 50% of the lung area was affected. The significantly higher lung pathology at 4 DPI infected tissues than at 14 DPI, which is indicating the self-limiting nature of this disease in hamsters. Immunofluorescence staining revealed viral antigen-positive bronchial and alveolar epithelial cells at 4 DPI, which were not detected at 14 DPI tissues. Immunoblood analysis of the lung tissue protein lysates support these findings. The viral genome copies number estimation showed the presence of viral genome in both 4 DPI and 14 DPI infected lung tissues. However, the copy number was significantly lower in 14 DPI tissues compared to 4 DPI tissues. For the quantitative proteomics analysis, all the differentially expressed proteins are presented as a heat map using hierarchical clustering analysis to compare mock-infected and infected samples, which is seen in figure A. 1,585 proteins identified across all samples. At 4 DPI, 50 proteins showed differential expression. From that, 33 upregulated and 17 downregulated. At 14 DPI, 18 proteins were differentially expressed, 9 upregulated and 9 downregulated. Principal component analysis revealed the distinct protein patterns between mock-infected 4 DPI and 14 DPI infected samples, which is seen in figure B. The pathway analysis was performed using online tool using Key-GGIP database. The enrichment analysis was performed for upregulated and downregulated differentially expressed proteins for all the 3 groups.
Based off the results, there are a few concluding points to be made about this study. The study observed abnormal expression patterns of surfactant proteins in lung tissues at both 4 and 14-day post-infection compared to mock-infected samples. This suggests a disruption in the usual functioning of lung respiratory gas exchange due to the viral infection. Previous reports have indicated that viral proteins may influence surfactant metabolism, which could potentially compromise the host's immune response. Additionally, we noted distinct damage to the epithelial-endothelial barrier in the 4 DPI infected lung tissues, which contributes to other respiratory viral infections and leaks blood components into alveolar spaces and lung tissues. In the proteomic analysis of lung tissues, it's noted that there is a decrease in certain protein levels in tissues with high lung pathology scores at 4 DPI. This indicates that certain proteins may have varying expression levels in the bloodstream compared to the primary site of infection, which is the lungs. This aligns with previous reports and supports the model's clinical relevance. The dysregulation of coagulation cascades noted the presence of 6 upregulated proteins in the study at 4 DPI compared to mock-infected groups. However, albumin and transferrin, which were downregulated in COVID-19 severity, were found to be upregulated in 4 DPI infection groups. Analyzing the profiles of serum samples from COVID-19 patients revealed dysregulation of pathways related to platelet and macrophage function. Efforts to understand these changes demonstrated that SARS CoV-2 infection enriches proteins associated with oxidative stress and inflammatory responses. The upregulation of oxidative expression in infected hamster lungs suggests an enhanced antioxidant response to prevent these stresses and damages to the lungs. Specific proteins such as thymosin beta-4 and hemoglobin 1 emerged as potential therapeutic targets for drug discovery in COVID-19 treatment. You might be wondering why these are important. Well, these 2 proteins are involved in wound healing and angiogenesis, which means the development of new blood vessels. They also help to break down blood clots and exhibit anti-inflammatory effects, which can help the immune response have more control. In conclusion, we will highlight limitations and future directions of this research. For limitations, the study acknowledges that the sample size and certain factors used for analysis were considerably small, which could potentially limit the generalizability of the findings to a larger population. The study also used a single viral dose, local isolating strain for the animal, which may not fully capture the variability encountered in real-world scenarios. There is also incomplete genome information of Syrian golden hamsters which could impact the findings. Furthermore, there is possible discrepancies in proteomic analysis between infected hamsters and humans due to differences in lung and blood proteomes, as well as limited hamster-specific reagents and antibodies that influence the depth of the analysis. Regarding future directions for this research, an expanded sample size and condition could provide comprehensive understanding of SARS-CoV-2 infection dynamics. Genome sequencing of the hamsters could provide genetic factors that may impact responses to the virus, as well as therapeutic interventions such as antiviral drugs could identify treatments to treat COVID-19. These potential future directions highlight the ongoing nature of research in this area and the need for continued exploration to deepen our understanding of SARS-CoV-2 infection and its impact on host organisms. Thank you for listening to our presentation.